Right now, before your first bite, your body is already getting ready to eat. One whiff of pizza, and your mouth starts to water, while your stomach begins preparing gastric juices. But the real danger is still ahead. In a couple of seconds, every bite you take will pass millimeters from the entrance to your lungs. One glitch in the timing, and you could start coughing. In a more serious case, some of the food can slip into your airway. So why don't you choke every single time you eat? What flips your throat from breathing to swallowing in a split second? And why does it all start in the brain, long before you swallow anything? Let's follow the journey of a single bite, from the brain's first signal to the moment the entrance to your lungs seals shut. Picture a lemon sitting in front of you. You slice it in half and juice beads up on the flesh. You bring one half to your mouth and touch it with your tongue. Maybe while you were imagining that, your mouth produced a little more saliva, even though there's no lemon around. This is the first secret of digestion. It can begin before food ever reaches your mouth. When you see pizza and catch its smell, odor molecules reach your nose, activate receptors, and signals travel to the brain. The brain recognizes food. It doesn't know the exact makeup, and it can't measure proteins and fats from a distance. It does something else. From past experience, it predicts what's coming next. It's like a weather forecast. There's no rain yet, but from the dark clouds, you already know it's time to shut the windows. For your body, the smell of food is like those same dark clouds. These prep reactions to the sight, smell, and even the thought of food are called the cephalic phase. And what matters isn't just the smell itself, but your experience with it. The aroma of freshly baked bread can awaken your appetite. But if a similar dish once made you sick, that same smell can trigger disgust. The smell didn't change. What changed is the meaning your brain gave it. This is exactly what Pavlov's famous experiments showed. Dogs started drooling in response to a signal that simply came before feeding. Their bodies took it as a promise of food. When the brain recognizes such a promise, the first commands go out to the organs, along the vagus nerve to the stomach and pancreas, along other paths to the salivary glands. The most noticeable reaction in the mouth. There's more saliva. But saliva doesn't just moisten your food. It helps gather it into a lump, delivers molecules to your taste receptors, and contains amylase, an enzyme that starts breaking down starch. At the same time, the stomach gets ready. Nerve signals increase the activity of the cells that produce gastric juice. The pancreas gets ready too. It'll need to send in bicarbonate and enzymes. In some people, there may even be a small early release of insulin before blood glucose ever rises, but this doesn't always happen. So the claim that one glance at dessert is bound to spike your insulin is overblown. Why all this in advance? Because these reactions take time. To produce saliva, increase digestive secretions, and coordinate several organs, it's better for the body to sometimes prep for food that won't come than to be late every time. So appetite doesn't start only with an empty stomach. Sometimes it starts with a forecast. The brain builds from a single smell, and now you take your first bite. Chewing seems like just prep work for digestion. But in fact, digestion has already begun. Teeth grind up food, and it's not only about making it easier to swallow. Picture an ordinary cube of bread. While it's whole, enzymes can mostly work along its surface. But crush that cube into dozens of tiny bits, and the total surface area shoots up. Enzymes gain access to a much larger surface area. At the same time, your tongue mixes the food with saliva, moistens it, and gathers it into a soft, cohesive lump, the bolus. And amylase is already taking apart starch while the food is still in your mouth. That's why the rule, chew every bite exactly 32 times, has no universal meaning. The body doesn't count jaw movements. It goes by the state of the food, whether it's become fine, moist, and cohesive enough to swallow safely. Yogurt barely needs chewing. A ripe banana falls apart on its own, but a nut or a raw vegetable takes far longer to break down. Their nutrients are hidden inside tough plant cells, and you still have to get to them. And now, the most dangerous moment of the whole journey. The swallowed lump has to pass through the pharynx. It's a shared stretch where the paths of food and air meet. Just below, they split. In front, the entrance to the larynx, leading to the trachea and lungs. behind the esophagus, to the stomach. Every bite passes right up against the entrance to your airway, and it must not go in there. While you chew, everything is under conscious control. But then your tongue pushes the lump back. The pharynx's receptors notice it, and an automatic program kicks in. Stopping a swallow that's already begun is nearly impossible. From the brainstem, commands fan out almost at once to the tongue, pharynx, larynx, and esophagus. First, breathing pauses for about half a second to a second and a half. This pause is called swallowing apnea. It keeps you from breathing and swallowing at once. Next, the vocal folds close, the entrance to the larynx narrows, and the larynx itself rises and moves forward. And the epiglottis tips back and helps steer the lump around the airway. The epiglottis is often called the lid that closes off the trachea, but that's too simple a picture. A safe swallow relies not on one lid, but on several layers at once. And the closed vocal folds especially matter. At the same time, the entrance to the esophagus opens, allowing the bolus to pass through. Then the gate returns to its usual mode, and breathing continues. Usually the swallow is tucked into an exhale. You breathe out. Take a short pause for the swallow, and breathe out again. If a crumb is left near the entrance to the larynx, the airflow points outward, not inward. That's an extra layer of protection. But sometimes the sequence gets thrown off, especially with liquid. It spreads faster than a dense lump moves and can reach the entrance to the larynx sooner. Then the backup defense kicks in, coughing. A sharp blast of air pushes the particle out. So a cough after a bad swallow doesn't mean the food reached your lungs. More often, the body reacts sooner than that. And now it's clear why we choke so easily when we talk or laugh while eating. For speech, the path through the larynx has to be open. For a swallow, it's the opposite. Closed. The body has to reconcile incompatible tasks in one brief moment. If everything went right, the lump ends up in the esophagus, and its walls drive the food down in waves, peristalsis. Food reaches the stomach not by gravity. The muscles do the main work. Swallowing isn't food falling down a tube. It's an instant flip of the gate between breathing and digestion. The lump has cleared the dangerous crossroads and is heading down the esophagus. Ahead, the stomach. But there, it's met by acid that can take meat apart. And here's the strange thing. The stomach itself is made of living tissue. So why doesn't it digest itself? Let's start with this. The stomach is not a motionless bag filled with acid at all. People often picture the stomach as a motionless bag, as if food just sits in the acid while it dissolves. But the stomach is moving all the time. Waves of muscle contractions travel along its walls and push the contents down toward the exit into the small intestine, the pylorus. And this exit doesn't work like an open door. During digestion, it stays very narrow. Liquid and small enough particles pass through, while larger fragments are held back. When a wave pushes the contents toward the nearly closed pylorus, a big part of it is squirted back into the stomach. This is called retropulsion. Strong contractions and jets of fluid mix and grind the particles again. Then the contents head for the exit once more and get pushed back again, if the pieces are still too big. So you get a kind of mill with a narrow gate. Usually solid particles have to shrink to about 1 or 2 millimeters before they can pass through. It's not an absolute rule. A lot depends on the structure of the food. But that's the basic principle. Little by little, the stomach turns food into a thick semi-liquid mass, chyme, and sends it into the intestine in small portions. And this is where it becomes clear why you really should chew carefully. The stomach can finish grinding what wasn't chewed enough, but it can't fully replace your teeth for every kind of food. Nuts and seeds have it especially hard. They're nutrients hidden inside dense cell walls. If the walls are intact, it's harder for enzymes to reach what's inside. But this moving acidic mix is constantly rubbing against the living surface of the organ. So why doesn't the stomach digest itself? First, let's figure out what actually breaks the food down. The cells of the stomach release hydrochloric acid. It creates a very acidic environment, changes the spatial structure of proteins, and activates an enzyme. But the acid doesn't do all the work by itself. The molecular scissors here are pepsin. It cuts long protein chains into short fragments. So the acid sets up the conditions, and the enzymes do the cutting. And here's the first problem. If pepsin cuts proteins, why doesn't it damage the very cell that made it? Because the cell releases an inactive precursor, pepsinogen. It turns into active pepsin only after it reaches the acidic cavity of the stomach. That's the first safeguard. The enzyme is produced in a safe, inactive form and switches on only where it's meant to work. But now acid and active pepsin sit right next to the entire inner surface of the stomach. What protects it? The first line is mucus. It coats the cells and slows the acid moving toward them. But mucus isn't an airtight film, and small hydrogen ions can pass through it. So inside the mucus layer, bicarbonate is held in place, able to neutralize the acid. Thanks to it, right at the cell surface, the environment becomes far less acidic than in the center. And normally it can get close to neutral. Across a thickness smaller than the mucus layer, two different chemical worlds exist: an acidic reactor in the center and a protected surface near the cells. And this barrier can't be built once and forgotten. Mucus is worn away along with the contents. So the cells constantly release new portions of mucus and bicarbonate. And what if some ions do break through? Under the mucus, they meet a dense layer of cells, joined so that the contents can't slip between them deeper into the tissue. And if a cell is damaged after all, neighboring ones quickly shift over and close the gap. And then dividing cells replace the damaged ones. Repairs like that need oxygen and nutrients, and the blood flow brings them. This defense is managed in part by prostaglandins. They support both the blood supply to the lining and the production of mucus with bicarbonate. So you get several levels of defense at once. Pepsin in its inactive form, mucus. Bicarbonate at the surface, tight junctions between the cells. Blood flow and constant repair. If one level lets a threat through, the next one meets it. But this system isn't absolute. If the balance between the aggressive environment and the protection breaks down, acid and pepsin really can damage the lining. For example, the bacterium H. pylori can cause chronic inflammation and weaken the protection. Another example, common painkillers, the NSAIDs. They suppress prostaglandins, and because of that, the lining may produce less protective mucus and bicarbonate, while its blood supply and recovery get worse. This doesn't mean every pill will cause an ulcer, but the principle matters. It's not only about the amount of acid. Sometimes the damage begins because the protection has weakened. So an ulcer isn't just a spot burned through by acid. It's the result of a broken balance between aggressive contents and the system that's supposed to keep them apart from living tissue. And that answers the main question. Food doesn't release mucus and bicarbonate, doesn't maintain blood flow, and doesn't repair its own surface. The stomach wall does all of that. The stomach protects itself not because the acid is harmless to it, but because every second it separates the aggressive contents from living tissue and constantly repairs minor damage. Now the stomach sends chyme into the intestine in portions. But after a fatty meal, the exit slows down noticeably. It feels as if the fat is heavy and gets stuck. In reality, fat doesn't stick to the stomach. Its muscles are perfectly able to move this food. The real break is farther along, in the small intestine. When the first portion of fatty chyme passes the pylorus, specialized cells of the lining recognize the products of fat digestion. The intestine doesn't know the name of the dish. It detects specific substances and uses them to gauge what kind of load has arrived. In response, hormonal and nervous feedback signals kick in. One of the main players is the hormone cholecystokinin, CCK. It helps slow gastric emptying, triggers the gallbladder to contract, and stimulates the release of pancreatic enzymes. But CCK doesn't act alone. Nerve reflexes and other intestinal signals work alongside it. Control is shared among cells, nerves, and hormones. The point of it all is the same. The stomach has to briefly reduce the delivery, while the intestine neutralizes the acid, mixes chyme with bile and enzymes, and processes what's arrived. At the same time, the intestine begins preparing the fat for digestion. The intestine's movements disperse it into tiny droplets, and bile salts coat them, stabilize them, and keep them from quickly merging back together. So the enzyme lipase gets more surface to work on. To coordinate all of this, a controlled flow is exactly what's needed. So the holdup of fatty food isn't a sign that the body can't cope. It's a way of not sending the next portion faster than the intestine is ready to take it. And that's exactly why fullness lasts longer after fatty food. The stomach stays stretched for longer, and the intestine keeps signaling the nervous system about the substances that arrived. But the emptying time can't be judged by the amount of fat alone. It's affected by the meal's volume and calories, the particle size, the ratio of proteins to carbs, fiber, acidity, and individual motility. More precisely, all else being equal, fat and a high-calorie content usually slow gastric emptying. The stomach pushes food forward, but the intestine sets the speed. Chyme moves on in portions. To process it, the pancreas releases enzymes, including proteases capable of cutting protein both in food and in your own cells. So why don't they start digesting the gland itself? And on top of that, the same gland explains a strange thing: why unsweetened bread can turn sweeter as you chew it. Let's start with the factory of dangerous tools. After a fatty meal, the stomach sends chyme into the gut in small portions, and the pancreas gets to work. It releases bicarbonate, which neutralizes the acid, plus a whole set of enzymes. Lipase breaks down fats, amylase works on starch, and proteases cut protein molecules apart. But the gland itself is made of cells and proteins. So why don't the enzymes that can destroy meat or an egg start digesting the very organ that made them? The most powerful protein-cutting scissors are produced mainly in an inactive form. Not all enzymes are stored inactive. Amylase and lipase can be released ready to work, but they act on carbs and fats. The main threat to cell proteins comes from proteases. That's why many of them are first built as inactive precursors, zymogens. Trypsin is made as trypsinogen, chymotrypsin as chymotrypsinogen. Until these molecules are activated, their scissors stay closed. But the protection doesn't stop there. The enzymes and their precursors are packed into separate containers, zymogen granules, each one sealed off by a membrane from the rest of the cell. So you get a double defense. The dangerous proteases are still switched off, and they're also isolated in transport containers. When the signal comes, the granules move to the cell surface. Their contents are dumped into a duct and travel through the duct system into the duodenum. And that's exactly where the scissors get switched on. On the surface of the gut cells, there's an enzyme called entropeptidase. It turns part of the trypsinogen into active trypsin. That's the first turn of the key. Then active trypsin starts turning new trypsinogen molecules into trypsin and switching on other proteases. A cascade begins. One activated molecule triggers the next ones, and those keep the process going inside the gut contents. In a few steps, a set of inactive precursors becomes a fully functioning protein digestion system. The gland makes the tools, but the command to switch them all on arrives right next to the food. Biological systems, though, aren't error-free. Sometimes a small amount of trypsin can activate prematurely. For that case, there's one more layer of protection: a trypsin inhibitor, known as SPINK1. It can bind and block part of the trypsin that switched on early, before it sets off a chain reaction. It's like an emergency fuse inside the container, but its capacity is limited. It can't neutralize an unlimited amount of trypsin. If storage, transport, or control break down, the enzymes can activate in the wrong place. Damage cells and trigger inflammation, pancreatitis. It's sometimes loosely called self-digestion, but the real mechanism is more complex. Along with premature activation, there are disrupted calcium signals and mitochondrial function, cellular stress, and an inflammatory response. So the gland isn't protected by one impenetrable shell. Its safety is built from a sequence of barriers: inactive precursors, isolation in granules, export through the ducts, an activation cascade only in the gut, and an inhibitor in case of an error. It doesn't make the proteases safe forever. It keeps them switched off and separates the place they're made from the place they're activated. Now the scissors are on and cutting protein, but right nearby another enzyme is working: pancreatic amylase, which breaks apart the starch from bread, potatoes, and rice. And here a familiar paradox shows up. If you chew plain bread for a while, its taste slowly turns sweeter. Where does the sweetness come from? If we didn't add anything to it, it didn't appear out of thin air. Starch is made up of long chains built from repeating units of glucose. Picture a necklace made of a huge number of identical beads. As long as they're joined into a long chain, the whole thing behaves differently than separate beads. It's a similar story with taste. Free molecules of some sugars taste sweet, while the large structure of starch is barely perceived as sweetness. The breakdown starts right in the mouth. The teeth grind the bread, the tongue mixes it with saliva, and salivary amylase starts snapping the bonds inside the starch chains. But it doesn't take the links off one by one. It acts like scissors, cutting a long necklace into several short fragments. Among them appear maltose, maltotriose, and other small chains. Some of them taste sweeter than the original starch. That's why the taste of bread changes as you chew. But in the mouth, the work is only beginning. After you swallow, amylase keeps acting in the stomach for a while, until the acid penetrates deeper, after which its activity drops sharply. The real continuation is in the small intestine. The acidic chyme mixes with pancreatic bicarbonate, the acidity drops, and pancreatic amylase keeps shortening the chains. But even it doesn't break starch all the way down to single glucose molecules. The final scissors sit on the surface of the gut cells, on the brush border. Here, the short chains meet maltase glucoamylase and sucrase isomaltase, which cut the remaining bonds, including the branch points. Only now are the individual glucose links fully released, and even free glucose is still inside the gut cavity. To get into the body, it has to pass through the gut cell. On the gut side, the transporter SGLT1 grabs it together with sodium and pulls it in. On the other side, the transporter GLUT2 lets it out further, into the blood heading to the liver. Even so, not all of the bread turns into glucose. It has proteins, water, fiber, a bit of fat, and some of the starch may turn out resistant to digestion and pass on through. The speed of breakdown depends on how finely the grain is milled, the cooking, and the amount of fiber. That's why different kinds of bread are digested differently. Bread doesn't turn into sugar by magic. Digestion simply takes apart the glucose chain hidden inside it. So the starch is taken apart, and glucose slips easily into the blood. It dissolves well, but protein and fat don't get inside that easily. And protein is tied to the big gym myth. Is it really true that chicken goes straight to your muscles? So how does food even cross the wall of the intestine? And why does that wall let the good stuff through, but hold back bacteria? Let's start with protein, and with that very gym myth. Picture this. You eat a piece of chicken after a workout. It seems logical that the protein gets digested, reaches the blood, and heads straight to your muscles. But not quite. A protein is a long chain of amino acids, folded into a complex three-dimensional structure, and its shape determines its properties. The protein in chicken, an egg, or beans was built by another organism for its own needs. Human cells can't just slot it in as a ready-made spare part. Besides, a whole molecule is usually too big to pass through the intestinal wall. So the body first takes the foreign structure apart. The first stage is in the stomach. The acidic environment changes the three-dimensional structure of proteins. They start to unfold, and this process is called denaturation. Picture a tightly tangled ball of yarn slowly coming undone. Hidden parts of the chain become more accessible. But the acid itself doesn't break protein down into amino acids. It creates the conditions for the enzyme pepsin. And pepsin works like molecular scissors and cuts the long chains into shorter fragments, peptides. Next, the partly disassembled protein moves into the small intestine. The acid gets neutralized, pepsin loses its activity, and the pancreatic proteases take over, the very ones that were stored switched off, so they wouldn't damage the place where they're made. Different enzymes attack different parts of the chain, and gradually free amino acids appear, along with short fragments of 2 or 3 amino acids. At the surface of the intestinal cells, single amino acids move in through special transporters, and dipeptides and tripeptides have a path of their own. But even these short fragments usually don't head into the blood hole. Inside the cell, enzymes more often cut them down to individual amino acids. Now the former structure is gone. For the cells, these are no longer amino acids from chicken, from an egg, or from beans, but universal building blocks. They exit the other side of the intestinal cell, enter the capillaries of the villus, and travel through the portal vein first of all to the liver. The liver uses some for its own tasks and regulates how they flow onward. After absorption, the amino acids enter a shared dynamic pool, a kind of distribution center, where the parts are constantly drawn out and used up. This isn't a warehouse of spare protein. The amino acids are in constant motion. From them, cells build enzymes, receptors, transport proteins, components of the immune system, skin, and the intestine. Some really does reach the muscles and goes into muscle protein synthesis. But the muscles don't get all of it. The intestine and liver take their share. The rest is distributed among the tissues. And if some amino acids aren't needed, the body can remove their nitrogen-containing group and send the rest into metabolism. So the protein you eat doesn't head straight to your muscles. First, it stops being the food's protein, gets taken apart into pieces, and only then does the body decide where to use them. But protein isn't the only thing the body first takes apart, then puts back together. Picture a drop of oil in a glass of water. No matter how you stir, after a while the oil gathers back at the surface. Fat barely dissolves in water, and inside the intestine it's a watery environment. And blood and lymph are mostly made of water. So how does fat get in? The body changes its form several times. First, the movements of the intestine physically break large fat droplets into many small ones, giving the enzymes access to a larger surface area. But small droplets tend to merge back together. Here bile salts help out. Each such molecule has one side drawn to fat and the other to water. They coat the droplets and keep them from merging. They don't cut the fat molecules. Their job is to keep many small droplets apart. Then pancreatic lipase joins in. Most dietary fat consists of triglycerides, like a backbone with 3 fatty acids attached. Lipase snips some of the bonds and turns them mainly into free fatty acids and monoglycerides. But even these products interact poorly with water and can't travel freely through the intestine. So bile salts create the next transport form, mixed micelles. A micelle isn't a sealed capsule, but a small dynamic structure, a kind of molecular ferry. It carries the fatty cargo to the wall of the intestine, but doesn't itself go into the cell. At the membrane, the fatty acids and monoglycerides leave the micelle and move inside, while the bile salts stay outside and get used again later. You'd think the fat could now go straight into the blood. But the cell does almost the opposite. It reassembles triglycerides from the pieces it received and packs them together with phospholipids, cholesterol, and proteins into a particle, a chylomicron. Why take apart, then build again? Large molecules were hard to move across the membrane. After disassembly, their pieces could cross the barrier. And now, from them, you can assemble a transport particle fit to travel through the body. But chylomicrons are too big for most capillaries of the villus, so they take a different path. In the center of the villus, there's a lymphatic vessel. The chylomicrons enter it, move through the lymphatic system to the thoracic duct, and only then does the lymph pour into a large vein, and the fat finally ends up in the blood. So most long-chain fat gets there not directly, but by a lymphatic detour. There's an exception. Short and medium-chain fatty acids interact better with water and can go into the blood more directly, toward the liver. Glucose, amino acids, and fat still have to cross the border between the contents of the intestine and the body's internal environment. And the intestine is not a sponge. Picture a glucose molecule right at the wall. Between it and the internal environment stands the intestinal epithelium, one cell layer thick. Until the molecule has crossed it, the body hasn't gotten it yet. The barrier's task seems almost impossible. Let water and nutrients through, but hold back bacteria and large structures. For this, the surface is organized into folds, villi, and tiny projections called microvilli. Together, they create an enormous contact area. But the larger the surface, the longer the border you have to control. Our glucose passes through a mobile layer of mucus. In the small intestine, it isn't a solid wall, or it would get in the way of absorption and reaches the membrane, where a transporter grabs it along with sodium. Now it's in the cell, but not yet in the blood. An intestinal cell has 2 different sides. On the intestinal side, transporters that take substances in. On the opposite side, systems that release them into the tissues. This creates a directed movement. Glucose enters on one side and exits on the other, into a capillary toward the liver. Such a path through the cell is called transcellular. And what about the bacterium nearby? There's no suitable transporter for it, and it's too big. It can't slip between the cells either. They're joined by tight junctions. But this isn't fixed cement. It's a regulated filter. It lets through water, ions, and some small substances, depending on their size, charge, and the state of the barrier. This is the paracellular path. The intestinal barrier works not because it's sealed shut, but because its permeability is selectively regulated. Normally, whole bacteria don't cross an intact epithelium, and beneath it the immune system is always on duty. It doesn't wait for a breakdown. It calmly guards the border and helps the body not react to ordinary food and a normal microbiota. But the barrier isn't invulnerable. With infection, inflammation, or cell damage, its permeability can change. And then substances that usually stay in the lumen cross the border. This doesn't mean every bit of bloating is caused by a leaky gut. Permeability is a regulated physiological property, and its disruption can't be diagnosed from a single nonspecific symptom. So glucose, amino acids, and fat cross the barrier, each by its own route, but some food is out of the body's reach entirely. Fiber, for example, your enzymes can't break down. So why do the bacteria in the large intestine finish eating for you? And why doesn't lunch have a single final destination? Let's start with what your own enzymes couldn't digest. Imagine you eat an apple. Its sugars are released during digestion, cross the intestinal barrier, and enter the blood. But part of the apple takes a different route. It's a fragment of the cell wall, one of the structures we call fiber. Human enzymes barely change the molecular structure of many kinds of fiber. So it reaches the large intestine. Along the way, the fibers can swell, bind water, and change their physical properties. But many chemical bonds inside these structures stay out of reach of our enzymes. It's not that the fiber is too tough. It's just that breaking a particular chemical bond requires the right enzyme, a tool whose shape matches a specific spot on the chain. And humans don't have the full set of these tools. It seems like the apple fragment will now just keep going and leave the body. But in the large intestine, it's met by a huge community of microorganisms. Different bacteria have genes that let them build enzymes that human cells don't produce. One bacterium latches onto the plant fiber and starts cutting the complex chain into shorter fragments. But on its own, it does only part of the job. These fragments get picked up by other microbes. Some keep taking them apart. Others turn the intermediate products into new substances. So the microbiota doesn't work like a single factory, but like a network of interconnected production lines, where the product of one microorganism becomes raw material for another. This process is called fermentation. Bacteria use fiber first of all for their own metabolism. They get substances and energy, grow, build new cells. Along the way, gases and many other products appear. And among them, 3 small molecules matter especially: acetate, propionate, and butyrate. They're called short-chain fatty acids. They're nothing like the big droplets of dietary fat, which needed bile, micelles, and chylomicrons. These molecules are much smaller and interact well with a watery environment. So some of them are absorbed by the cells of the large intestine. Butyrate is used especially actively by the gut lining cells themselves. For them, it's an important source of energy. Some of the propionate and acetate goes into the portal blood to the liver. The liver actively uses propionate, while some of the acetate can pass into the general bloodstream. So you get an unexpected route. Human enzymes couldn't take apart the plant fiber, but bacteria turn part of its structure into small molecules, which the gut is already able to absorb. That said, this doesn't happen with all fiber. Fiber isn't one substance, but many structures with different properties. Some fibers are actively fermented, others only partially, and some pass through almost untouched. The result also depends on the makeup of the microbiota, and that's different for everyone. So the same fiber is processed differently in different people and yields different amounts of fermentation products. This doesn't mean one person has good bacteria and another has bad ones. The microbiota is a changing ecosystem whose activity depends on the available raw material, interactions between microbes, and conditions in the gut. Meanwhile, our fragment of apple wall has split up. Some of its atoms became part of bacterial cells. Some ended up in gases and other fermentation products. Some atoms became part of acetate, propionate, or butyrate, and the unfermented remains continued on through the large intestine. So bacteria don't give the person all the energy from fiber. They use it for themselves, but some of the products they make become available to the host body too. Where human enzymes end, the chemical work of the microbiota begins. Now lunch's journey is almost complete. Glucose has ended up in the blood. Fat has passed through the lymph. Amino acids have spread out among the tissues. Even part of the fiber, thanks to bacteria, has turned into new molecules. But what will happen to all of this next? A few hours ago, lunch was sitting in front of you. Bread, meat, vegetables, and a bit of oil, all on one plate. Now that dish no longer exists as a single whole. Starch is broken down into simple sugars, proteins into amino acids. Most of the fat is packed into transport particles, and the fiber is moving through the gut, or has already gone to the microbiota. After absorption, the molecules arrive at a metabolic crossroads. They can be used right now, built into new structures, stored for a while, or gradually removed. And these aren't four separate final destinations. The very same molecule can, over time, go down several routes. Let's start with glucose. Through the portal vein, it goes to the liver, an important distribution hub, which can hold back part of it, change its route, or let it pass on into the general bloodstream. But glucose doesn't turn into pure energy. Inside cells, it goes through a chain of chemical reactions, and part of the energy locked in it is transferred onto ATP molecules, a universal carrier of readily available energy for many cellular processes. The atoms of the glucose itself don't simply disappear. With full oxidation, its carbon ends up as part of carbon dioxide. The blood carries it to the lungs, and some of the atoms of your recent lunch leave the body with your breath. And glucose doesn't have to be spent right away. It can be set aside for later in the form of glycogen. Liver glycogen helps keep the blood sugar level steady between meals, while muscle glycogen is used mainly by the muscles themselves. Dietary fat has its own route. Most long-chain fats leave the gut inside chylomicrons, pass through the lymph, and enter the blood, where a special enzyme helps release the fatty acids. Some tissues use them as fuel. Others build them into cell membranes and signaling molecules. The excess can be stored for a while in fat cells. But even that isn't the final stop. When needed, the store is taken apart again, and the fatty acids return to the bloodstream. Amino acids don't head to one place either. Some are used first by the gut and liver. The rest are distributed among the tissues and go toward enzymes, receptors, antibodies, and renewal, including of muscle proteins. The body has no separate storehouse for spare amino acids. If their carbon part is used in metabolism, the nitrogen has to be removed. In the liver, it's turned into urea, which enters the blood, is filtered by the kidneys, and leaves with the urine. Not everything that was on the plate manages to go through these routes within a few hours. What's undigested keeps moving through the gut. Part of the fiber is processed by the microbiota. Water and some of the fermentation products are absorbed, and the rest leaves the body later. That's why lunch doesn't have a single final destination, and a few hours isn't a strict physiological deadline. The speed depends on the makeup and size of the portion, the work of the stomach and gut, and many individual factors. What to take from the finale? Fiber isn't there just to check a box. By fermenting it, bacteria produce butyrate, which directly feeds the cells of your gut. So varied plant food feeds not only you, but also your microbiota, and through it, the gut wall itself. And the second thing, what you eat doesn't have a single fate. A molecule's energy can be handed off to other processes, and its atoms can be stored for a while, built into new structures, or gradually removed. So the point isn't in one magic food, but in the overall balance of what comes in regularly. It all began even before the first bite. The brain noticed the food and started the preparations. The teeth ground it up. The throat switched the paths of air and food. The stomach mixed the contents and protected its own walls. The pancreas, bile, and intestinal enzymes took apart the nutrients. The intestinal barrier let the suitable molecules through, while the microbiota handled what human enzymes couldn't break down. The food's journey through the digestive system is almost complete. But the story of its molecules goes on. Food becomes part of you not because it stays inside whole. The body takes it apart, reassembles it, and distributes its atoms between the life of cells and the outside world. This was the season What Happens to Food Inside You. Subscribe to BioCode Lab, where the complex becomes simple.